Thank you all for joining us at YWIB Online. And for those of you, some of you may be viewing this, the recorded version eventually on Young Women in Bio on our YouTube channel. So I would like to thank you all for coming either to the live event or to the online event. And so my name is Christina Herbert, and I'm the chair of Young Women in Bio Southern California, which is based in the San Diego and Los Angeles areas. And this event is our second event of 3 experiential learning sessions being hosted by YWIB SoCal, and the sessions are named Genomes, Germs, and Jewelry, and this is part 2, so it's specifically named It's in the Genes. And so this event is being recorded on July 15th, 2020, and will be available on the YWIB online platform shortly or after this, probably at least by the end of the month, and it will be available for students across the United States and in Canada for free access to our educational and leadership programs and other opportunities in science, technology, engineering, and STEM. And so we're so excited to have you all here with us and to be conducting this summer series that seeks to bring all of the hands-on activities we normally have for students in Southern California, coupled with discussions with leaders in the biotech industry. And we normally bring these events live. But now, due to COVID-19, we have this new venue for bringing these events to people. And in reality, it's kind of a great thing to some extent in that we get to engage quite a number of students, but probably a lot more than we could at live events. So thank you for joining us. I just want to give you a quick rundown of what Young Women in Bio is all about. And so Young Women in Bio gives girls today the inspiration and support they need to become tomorrow's leaders in science, technology, engineering, and math, so otherwise known as STEM. And we strive to provide education and hands-on experiences in STEM, as well as share our passion for all the scientific fields. So Women in Bio, while the name says women and it says bio, we're not just women, there are men that join our group, and we do like to try and ignite curiosity in all fields of STEM, not just bio, although we do have a bit of a bio focus. So YWIB is launching this new initiative for young women who are curious about STEM and want to learn more so that they can participate and learn from events that are being put on in any of our 13 YWIB chapters in cities across the United States and in Montreal. And so the launch of YWIB Online comes at this time when there's a real need to provide young women who cannot otherwise attend our events and programs with access to online programming in STEM. So this will help with our goal to ignite curiosity and to inspire young women to see a future for themselves in STEM. So we believe that by making these online resources available, we can reach an even broader audience, even more young women, and help them to find a lifelong passion and career in STEM. So, as I said, YWIB supports young women nationally. We partner with all sorts of groups like leading companies, universities, hospitals, and other organizations around the country to host these engaging events. And in fact, in one year, we've had more than 70 events across North America, and we reached more than 4,500 young women and had the support of 250 volunteers. So with our YWIB Online program, we're hoping to reach even more students and more volunteers. And as I said, we have 13 YWIB chapters across the United States and 1 in Canada. And so if you want to learn more about Young Women in Bio, our website is right there, womeninbio.org/YWIB. And so I wanted to just briefly, before we start the formal part of the presentation, to tell you a little bit about an initiative that we are starting here in Southern California. This is going to be the Biotech Influencers Program, and we'll be launching this in the fall. And this program will be a monthly webinar series to discover your abilities in future in STEM. You'll have a chance to engage with thought leaders, innovators, and industry experts who aim to bridge the gender gap as you realize your full STEM potential. So you'll get to network, you'll get live group coaching with some of the leaders in the field, you'll earn a shareable completion certificate, and all of this will be provided free of charge. So you should look for a flyer starting probably at the beginning of August, and registration will begin late August, and the program will eventually launch in October. So, and this program is intended for high school students. Okay, and so with that, I wanted to get started with today's events, Genomes, Germs, and Jewelry. This is part 2, It's in the Genes. And our speakers for today are Dr. Beata Mierswa and Ashley Van Zeelen. And we will start with Dr. Mierswa demonstrating how to create a DNA necklace. And so this is really awesome. Like the beads are terrific. So Dr. Mierswa is a molecular biologist and an American Association for the Advancement of Sciences, if-then ambassador. And the ambassadors program brings together 125 women from a variety of STEM careers to serve as high-profile role models for middle school girls. And Beata makes an amazing ambassador for women in science as she does incredible research in cell biology. And now she's working at the Ludwig Institute for Cancer Research and UC at San Diego, and she's studying how different types of cells divide to broaden the world's understanding of cell division and improve cancer therapy. And you'll get to see how she's combined her passion for science with also her passion for art in her talk. And then after hearing from Beata, we'll hear from Dr. Ashley Van Zeelen. And Dr. Van Zeelen has been leading innovative genomics research and development programs for more than 15 years and is currently Vice President of Product Integration and Customer Collaboration at Illumina. So she was co-founder and CEO of Cipher Genomics, a cloud-based genome interpretation company. And that was sold to Human Longevity a while back in 2015. And she's been the recipient of numerous business and science awards, including the Biocom Life Science Catalyst Award, UCSD 40 Under 40, Emerging Leader Alumni Awards, and there's a ton of awards here. So she holds a PhD in neuroscience from UCLA. And so I can't wait to learn about the secrets of her amazing success that she's had in her career. And so she can tell you guys all how she got there. So thank you guys for joining us. Then I just want to thank also our sponsors. So New England Biolabs and Scientist.com are our local Southern California sponsors, and they help pay for all of the supplies that you have all hopefully received or will receive if you haven't gotten them yet, okay? And so with that, I would like to let Beata take over. I will stop sharing my screen and let her get started.
Okay, let me start my video. Can you hear me and can you see my title slide?
Yes,
okay,
we can.
Perfect, thank you. Great. So first of all, hi everyone. I'm really excited to see so many of you here, and I'm really excited to chat with you and create some DNA jewelry together. So my name is Beata, and as Christina already said, I'm a molecular biologist here in San Diego, and I do my postdoctoral research and I study how human cells divide. And so along my academic career path, like classical way of doing it, I also create science, art, and fashion. And I do this because I want to share the beauty of biology with the world and get people excited about science. So before we start with the jewelry, I want to briefly tell you a little bit about what I do. I know some of you are curious about what it's like to work in research and how I got to combine my passions for science and art. So I just want to give you a little bit of an overview of my journey, and we can talk about the details later. So I was born in Poland, in Europe, in a very small village in the middle of nowhere, so there were no scientists around. But I always loved art, and I was always curious about science, but it never even crossed my mind that I could combine art and science until much, much later. And so in school, when I was going to school, I used to make drawings for my friends, but I never really thought about it as a serious career path. And I didn't really know much about science either. I was fascinated by it, but I didn't know what a lab looked like from the inside. But I loved the concept of cells and DNA and understanding what we are made of. And so that's what inspired me to start studying molecular biology. So I really had no idea what to expect, but I was working on a lot of different things. And when I was doing my master's thesis, I started doing a lot of microscopy. So I was using all of these fancy microscopes to watch cells divide. And this is when I fell in love with cell division. So here's a movie that I quickly want to show you just so you get an idea of what it looks like. So this is a movie that I recorded at some point. And I was always fascinated with the beauty about, you know, how beautiful it is, but also about how much we still don't understand about it. And so ever since then, I've been working on different fields in cell division. And so I did my PhD project in Austria, and then I moved to San Diego, where I am now, and I study how different types of cells divide. And the aim is to improve the way we can treat cancer. And I know somebody was curious about what the best and the worst things are about working in a lab. So I just quickly wanted to touch on that because I think it might be interesting for some of you. So I love what I do because I get to use a lot of creativity. So I get to, you know, design my own experiments. I get to think outside the box. And I have a lot of freedom to play around with things and try things out. One thing that I really had to get used to was a big challenge for me was to figure out how to deal with experiments that don't work. Because the truth is most experiments fail. It's by definition, that's what it is, right? So you try out a lot of things and you probably need to try a few things until it starts working. So at the beginning, I kind of started taking this personally, and I'm sure a lot of scientists can relate to that, but it took me a while to internalize that this is part of the process. But then again, when I look through the microscope and I have the chance to see something beautiful and something nobody has ever seen before, that's when I get really excited, and that's what makes it all worth it. So yeah, so that was about my science. I want to tell you a little bit about my art as well. So up until halfway through my PhD, I had no idea that I could combine art and science. I always saw it as a separate thing. So for many years, I fully focused on working in the lab. And then one day I had the opportunity to make a drawing about my research topic at that time. So I was working on the final step of cell division, which is what you can see here. This is a real image of a human cell. And you can see that there's two cells almost already finished division, but you still have to split the bridge between them. So I was working on how this bridge is split at the final step of cell division. And so I made a drawing about this. So this is what I drew. It's two cells, just like above, and they're literally using scissors to cut each other apart. So I just did that for fun. I just wanted to see what it's like. And because I had the drawing, I started using it in my presentations. And then something amazing happened. So I noticed that people from my institute and people from my conferences, they would remember my research even years after I first presented it. And that's because they remember the drawing because it was something different. And through the drawing, they also remembered the science. So that's the first time I realized that science art could be a really great way to present my research and get other people excited about science as well. So since then, this inspired me to do a lot of experiments in terms of how I can communicate my research. And I started making drawings for my friends first, and then also for journal covers and conferences. So it was really amazing to start sharing my art online and get connected with like-minded people. And then I also started creating science fashion because I always loved wearing colorful clothes, and I always got to see all of these beautiful images, and I wanted to find a way to share them with the world and to get people curious and get people excited about science. So that's how I got to combine the two, and I love what I do now. I'm really happy that I was able to discover it. And so one thing I just want to say before you start making jewelry is that don't be afraid to try things out. If you love different things that you think you can combine, or maybe even science, art, and fashion, it doesn't mean that you have to pick one. You can love art, you can love creativity, and you can also be an amazing scientist. So definitely go for it and try it out. Okay, so let's switch to the DNA jewelry part. First of all, I want to give you a very, very short introduction about DNA. It's going to be very simple because I know we have a very diverse audience. So if you're an expert, you can just sit back and relax. And if you're new to this, I hope I can show you something and you can discover something amazing and fascinating. DNA, as you probably have heard, is material that carries all our genetic information. And it makes us who we are, basically. So our bodies are made of a lot of cells, and almost every single cell in our body contains our whole DNA. And DNA is like this long molecule that is shaped like a double helix. I'm sure you have seen this before. And it's made of, our genetic code is made of 4 different letters. And these letters are, the sequence of these letters is what contains the instructions of making all of the proteins in the bodies and what makes us who we are. So with the jewelry, we are going to replicate the structure of DNA. And so while we are making it, we can also learn about it. So let's take a closer look first. So DNA is made of 2 strands that pair with each other. And each strand has 2 parts. One of them is the backbone, which is this blue ribbon here that you see. And then the other part are the bases, the sequence of the bases, which are the colorful sticks here that connect with each other and connect the 2 separate strands. So when we take a closer look, the backbone is made of a sequence of sugars and phosphates. So they alternate when they're bound to each other, and that's what they look like in terms of the chemical structure. And so this is what holds everything together. And for the necklace, we're going to use 2 different types of round beads for the phosphate and the sugar. And then the second part are the DNA bases. So the bases here are lined up. So they bound to the sugars and they're lined up in a sequence. And you can see that there's 4 different types. There's guanine, cytosine, thymine, and adenine. And they are able to pair with each other. But they don't pair randomly. It's always like that guanine pairs with cytosine and thymine pairs with adenine. And as I said before, the sequence is what makes our genetic code. So I always find it fascinating to think about it, even though I've heard it so many times, that there's only 4 letters in our genetic code, but still it determines everything about us, like our hair color and how tall we are. And I just find it really fascinating to think about the concept of that. And one more thing, if you look closely at the bases, you can see that there's 2 different types that look similar to each other. So you can see that guanine and adenine both have this double ring structure, so they're slightly bigger. And then the cytosine and the thymine have a single ring. So when we are going to make our necklaces, we have the bases, the long beads for the bases, and some of them are slightly longer than the others. And so keep in mind that the longer ones are supposed to represent the guanine and adenine, and the shorter ones, the cytosine and the thymine. Okay, so if you have signed up and you have received the kit, you will have received all of the materials that you need for this. So you have the beads and the wire and everything. If you don't have the kit, or if you're watching this sometime in the future, you can go to my website, and on this page, there's a lot of things that you can find. You can find a list of materials that you will need, or you can get a kit that contains everything already together. And there's also a video that I created that contains all of the instructions so that you can follow along with it and take your time. So that's basically it. I hope you will enjoy it. I know you probably weren't able to follow along now, but if you sit down and finish it later on, I would love to see it, because a lot of you have different colors, and it'd be really great if you could share it if you want to. If you decide to share it, you can use some of these hashtags. And if not, basically, definitely check out SciArt and Science Fashion for inspiration. There's so many amazing science artists out there and so much amazing science, art, and fashion. So if you ever need some inspiration, those are good hashtags to check out anyway. And then you can, if you don't have a kit, you can go to the website. Again, here's the link. And you can find a materials list, or you can get your own kit, and you can look at the instruction video. So yeah, I hope that's it. I hope you enjoyed that. And please feel free to contact me anytime. On the bottom, there's my email address and website and hashtag for social media. So reach out to me anytime. I would love to get in touch, and I'm excited to discuss with you later. Thank you so much.
Okay, thank you, Beata. That was terrific. I know people had a little bit of trouble probably following along at the beginning, but if you follow the instructions or watch her video online, I think you can get through sort of once you get through the first couple steps, it's just really repetitive. So I think you guys will be able to do it. Just, try and try again. It's just like we said on the last event. Science is a bit about trying and trying again. Okay. So now I think Beata and I are going to turn our cameras off. And Ashley, if you'd like to get started, I'm excited to hear about your work. Thank you so much.
Thank you.
All right. Thank you so much, Beata. That was incredible. And I know I'm not the only one who desperately wants that beautiful gown that you showed, and those shawls and that art. It's just really, really incredible, beautiful stuff. So thank you. All right, let me pull up a couple of slides just to help me kind of talk through my journey and kind of what I do. So let me go back here. Okay. Now, are you seeing a bunch of smiling faces and Genomics 101, I hope?
Yes, we are.
Yeah.
All right, wonderful. Well, thank you also to Christina, and I see Alexa here as well, and Young Women in Bio for the invitation to talk to you tonight. This is, one of my favorite things to do. Actually, I love STEM. I love science. I would show you my DNA nerd shoes today if you're ever interested. Look up toms.com. So this is a treat for me. So I'll tell you a little bit about myself. I'll tell you a little bit about the company that I work at, Illumina, and then go into a little bit more about the genome and all the incredible ways that you can use genomics, what the future of genomics might look like, and then we can talk a little bit about careers or career paths if you're interested at the end. So introducing myself, as you heard, I'm a vice president in product development at Illumina. My area is responsible for product integration, meaning how we actually put the sequencing systems together, and those sequencing systems also have steps that happen upstream of them and downstream of them that we work to integrate as well. And then customer collaborations. So I'll share some stories later about how we've worked with leading scientists and researchers to create new applications of using genomic sequencing data to help patients with difficult diagnoses. As you heard, I've been to many schools, lots of different degrees, love education, love lifelong learning. So got psychology degree from University of Colorado, and that sparked my interest in human development, PhD in neuroscience, and then went to business school to kind of bring it all together and figure out how I can make an impact that way with science. From Colorado, live in San Diego here, love everything about the San Diego environment, surfing, fishing, etc. And so I was, like Beata, I was always a super curious kid, constantly dragging my parents around to see, when I wanted to be a paleontologist, we had to go to Dinosaur National Park. Wanted to be president, I started a government club. I mean, just always kind of very curious, wanting to explore the world around me. Following that natural curiosity, I really got interested, as I mentioned, in brain development. That got me interested in brain imaging. And I loved using the equipment, the MRI brain scanners. I actually also thought it was incredibly beautiful to be able to get those images of the brain and look at the function and the structure of those to understand biology and disease. At the time, I then started combining that imaging data with genomics data as well. And so how do you marry up information from mutations at the DNA level to what that might look like in brain imaging to understand disease biology? So that got me interested in kind of the whole computational side of how do we do this at scale? What are the right statistical methods? How do we mine information and data to do that? So I pursued a postdoc in statistical genetics and bioinformatics, and then just got, like I said, interested in how do you take all of that incredible innovation and turn it into products, and got interested in the business side. So all of that led me to take the opportunity to spin out this company, Cipher Genomics, which you heard was acquired by Craig Venter and a company called Human Longevity. And again, there I was responsible for overseeing a lot of the data platforms because, as you'll hear, your genome is made up of billions of those A, T, G, and C pairs. And so how do you sort through which one letter change out of billions is making somebody sick? It's a super hard problem. We are fortunate to be able to throw a lot of computation at it to do that. And so that's really what most of my career has been on. And then about 2 years ago, I had the opportunity to join Illumina in this role, and it's just been absolutely wonderful. So who is Illumina as a company? So Illumina is headquartered here in San Diego, founded just over 20 years ago, and today is about 8,000 employees with 22 offices globally. We build a lot of technologies, but are most known for the technologies that help us actually read out the base pair information, those A's, T's, G's, and C's that make up your DNA genome. So you can see lots of awards for how we do this, and it's been just a great company to work for. So I wanted to show a picture of what these systems actually look like. So here you can see kind of our portfolio of the actual sequencing technology systems, or the platforms, where we can put a biological sample on, and then what comes out the other end is all of that computational information that has, the A, T, G, and C in sequence order of your genome. And sometimes you want to look at the whole human genome of a lot of people, and for that you can use this big system here, the NovaSeq. Or you might want to look at, smaller genomes of bacteria or viruses, or, other smaller panels of information. And we have less expensive, smaller systems that kind of fit on a lab bench to do that. So as you'll hear, there are so many questions that we can ask about human health, about our environment, through understanding that core information of DNA. That we really have an opportunity to kind of address all of those with different kinds of products. And then I wanted to, in the spirit of today's talk, wanted to share the latest sequencing platform that we released just this year, which is this beauty right here called the NextSeq 1000 and 2000. So you may not totally appreciate this, but when you walk into a typical lab, you don't ever see instruments that look like this. So this is really, reflecting, I think, a different way of thinking about how that environment could be. So we have this beautiful light bar that changes color to indicate where you are in your sequencing workflow. Super slick user interface there. You can see that big start button and aluminum encased, like your MacBook. So incredibly beautiful instrument. Want to have a great experience, even if you're a scientist in the lab. I think many of us appreciate that beauty and that creativity at the intersection of business, of science and art. So a couple quick quizzes. You probably know this, I would guess. How much DNA do you, how much genomic information do you have in common with your neighbor? It is a whopping 99.9%. And so when you think about, out of all of that information, it's only 0.1% of our genetic makeup that makes any of us different, right? But even in that 0.1%, what a rich amount of diversity it offers. Everything, as you heard, from hair color and skin color to how tall you are to, your risk of diseases. So we're really focusing a lot of attention on understanding that small fraction that makes us unique. So as you heard in Beata's talk, what makes up a genome? Clearly, it's nestled right inside the nucleus of your cell. So if you have, 37 trillion cells and you burrow in to the nucleus of those, you start to unpack the genome. The genome, as defined, is just the full set of DNA in the nucleus. So for humans, that's 23 pairs of chromosomes. You have one copy from mom and one copy from dad. As you unwind those chromosomes, that's when you get to this beautiful double helix structure that you now can build and wear with pride. And that's so beautiful. So you know about the DNA building blocks. When you think about what sequencing a whole human genome means, you have to think about how many letters and how many nucleotide pairs there are in a genome. So the human genome is composed of 6 billion letters, because again, you get 3 billion from mom and 3 billion from dad. So when you really want to understand the full spectrum of any mutations that you might have inherited, you need to look at both copies. And so how do you think about how big 6 billion is? Well, if you were to publish a book with 6 billion letters all spelled out, you would fill 200 New York City phone books. So just a massive amount of text. And this is why when we sequence all of this information, I believe last year, we have about 15,000 installed sequencers. We generated over 150 petabases of data, of letters, 150 petabases. That's 500 years of continuous video streaming, right? It's multiple orders of magnitude over the Netflix catalog on an annual basis is the amount of genomic data we're creating because of how big this information is and how many letters you have. Another way to think about this is, okay, so how far away is the moon? It's about 238,000 miles away from Earth. If you took all the DNA from every one of your cells and stretched it out, that's about 6 feet of DNA packed in every little cell. So you have 37 trillion cells. So your genome unwound and stacked end to end would go to the moon 6,000 times and back, right? Or to the sun 600 times and back. So it's just really an incredible system that I think biology has really engineered in a fantastic way. So why do I get so jazzed up about understanding the genome and have really devoted my career to this? So as you heard, probably in previous talks and earlier this evening, it's really the blueprint of life, right? It really is the core software code upon which all of our systems develop. So it's the recipe. And just like any software code, it could have bugs in it and it can go wrong. And we want to understand that. We want to understand what in the genome makes you healthy and how do we encourage that in biology, and also what makes you sick. And so by really sequencing and deeply understanding that in many, many people, we can start to unpack those mysteries and develop new drugs. So some of the things that your genome can tell you, today is whether a medication will work for you or not. So some medicines you have to kind of metabolize in your body to make it an active compound. And if you have a mutation in a gene, you might not metabolize it so well. And so that drug won't work for you. Or you might metabolize it too fast and you don't give it a chance to act. So we know that information. Same thing with cancer treatments, right? You may have a mutation in your cancer that is particularly susceptible to a drug. And so we want to know that information and treat accordingly. If you're sick, we know there are disease predispositions in your genome, like BRCA1 for breast cancer is a commonly known one. And that's important information to manage your healthcare. Obviously, everything living has a genome. So how animals have evolved over time is important. The broader ecosystem, our kind of Earth's family tree, if you will, and your own family tree. So where did you come from? You might have heard of companies like 23andMe or Ancestry.com that can take your DNA and kind of really finely map back to, which parts of, Europe or Asia or islands that you have genetic remnants of. And then also agricultural applications. So we can look, just like there are genomes signatures that predispose humans to be healthy or sick, or stronger or more robust. Traits that are present like that in plants exist as well. So we can look at that and help farmers grow better crops. And then I'm sure you've all seen CSI or have heard on the news about forensic applications and use of DNA to kind of solve crimes, track criminals, really taken off in that space. So hold on, I might have to fix my blinds here so I don't get blinded. Okay, so how does genomics impact you? Well, as you heard, it expresses itself in how you look, right? So do you have curly hair or straight hair? One thing that I found interesting is whether you have dimples or not. Actually, a dimple, which I don't have, is a genetic mutation. So it actually means that you have shortened cheek muscles, and typically they're on both sides, but occasionally that rare one dimple is very cute. So that's hereditary. Whether you're right-handed or left-handed, how you taste, any number of really interesting, what we call phenotypes or kind of traits that are expressed from your DNA. So then genomics is just really broadly the study of your DNA, of the structure, of the function, of the evolution, of mapping these genes and where we can find these heritable traits. So the thing that I find just endlessly fascinating and compelling about devoting my career to this space is just, like I said, the breadth of applications that can leverage this information in a really impactful way. So the mission of the company, Illumina, is to improve human health by unlocking the power of the genome. And so we've talked through some of those applications, but what exactly does that mean? And how would Illumina as a company, really fulfill that mission? Well, the first thing that we can do is drive down the cost of doing this sequencing. So back when I was in graduate school, or the Human Genome Project when I was in college, was just wrapping up, and it took 10 years and $3 billion to sequence the first human genome. I remember it was a big deal. President stood on the Rose Garden and kind of declared a new era of medicine, and he was right. But you can see that at $3 billion, it's not really accessible to do large studies. Then the first individual genome was sequenced for about $20,000. And then we really got to the technology that Illumina is built on today in about 2007 that had a $2 million price tag. And that's when I started to get interested. I thought, well, geez, okay, so if it's already fallen this much, I can see where this is going. And I think it'd be really cool to understand more how to use this technology to answer my biological questions that I had as a researcher. And you can see the price continues to fall. And so today it costs about $1,000 to sequence a whole human genome. So from, in 20 years, from $3 billion to $1,000 is a pretty remarkable drop. And so now that you have something that's $1,000, what are you going to do with it? And so that's where, again, we go back to kind of all of the great scientific questions that you can ask with this genome sequencing capability. So everything from reproductive health to biopharma. So reproductive health, even you saw pictures, I have 2 little kids. And for both of my pregnancies, rather than having invasive amniocentesis, where they check the health of the baby by inserting a very long needle into the pregnant mom's stomach to get a sample of the amniotic fluid, there's enough circulating baby DNA in a pregnant mom's body that you can just take a blood draw from your elbow and read out the baby's DNA and look for major chromosomal abnormalities. So that has been an incredible advancement kind of overnight adoption for reproductive health, because it's just much safer for mom and baby to do it non-invasively with just a blood draw than the big needle in your tummy. Oncology, I talked about. For cancer, you can sequence tumors and find out what's causing those tumors to drive, and then match that up with the right drug or the therapy that works really well against that particular tumor. Research, consumer genomics, we talked about the ancestry mapping, forensics, agriculture, et cetera. So as we continue to drive down the cost of what it takes to unpack all of that information, the scientific community keeps coming up with more and more interesting new applications. Infectious disease, obviously, we're all facing the challenges with COVID, but SARS-CoV-2, the virus that causes COVID, was first sequenced on an Illumina platform in China by the Chinese CDC. That data was published, and within less than 2 days, a company had a vaccine candidate designed against that genetic data that they could start moving forward into trials. And so it really is an incredibly exciting time to be in the field, to be working on these technologies. And I just picture, you know, the opportunities that you will all have in this space, and it is just mind-blowing. So I wanted to end on just a couple of stories that really, I think, show the value of this that's happening today. So this isn't some future precision medicine vision. This is actually happening. So this is Joshua. Joshua lives in Wisconsin and went on a family trip to Puerto Rico. Coming back from Puerto Rico, he started having fevers and headaches, and it ultimately turned into seizures. And his doctor did a workup. They couldn't find out what was going on. And so for 4 months, he was battling this. You can see he finally went into a medical induced coma and heavy treatment to kind of reduce the brain swelling. So last ditch effort, they took a sample of the fluid that surrounds the brain and spinal cord and sent it to UCSF for analysis. They said, please try to find anything in here. So they sequenced it, and within 48 hours of receiving the sample, the lab at UCSF came back with an identification of leptospirosis in that cerebral spinal fluid, and they could treat with penicillin. And so he was discharged within a week. So 4 months of suffering, within 48 hours of sequencing the right sample, they had an answer. Another incredible story here in San Diego is this woman, Marin, a 39-year-old expectant mother, had the same prenatal test that I had, so non-invasive prenatal testing to look for baby's DNA, but it came back with some abnormal results. And so they did further follow-up testing, which you would do, and found out that fortunately the baby was healthy. However, what they found out was that that abnormal DNA readout was because Marin had tumor DNA floating around in her blood. So she actually had developed cancer that she was unaware of. And so after the baby was born, she was able to immediately start treatment for a previously undetected cancer. So this started a whole field of what's called liquid biopsy. And companies have spun up now to focus on how do you look for these circulating cancer DNA signatures in blood to do early detection or screening of cancers so that you can intervene right away. And then finally, this is Hassan, sweet boy born with an ultra rare disease, epidermolysis bullosa. Essentially, his skin is incredibly, incredibly fragile. So they say as fragile as butterfly wings. Almost half die before adolescence, and there's about 500,000 people affected with this disease worldwide. So by the time Hassan was 7, he had lost about 2/3 of his skin. And you can imagine the risk of infection and the challenges there. So what was pursued as a treatment is to take stem cells from a very small biopsy and actually correct the mutated gene that was causing this disease. So they corrected that gene in the stem cells and were able to grow out skin grafts that didn't have the mutated gene. And then they grafted that healthy skin back onto Hassan. And 18 months after surgery, he had, I think, 80% of his skin replaced. And you can see is smiling and happy and the outcome that every parent would want. So I hope that I've told you a little bit of why I've devoted my career to this, why I still get so excited to get up every day and go into work. And so, you know, it's not, I'm a neuroscientist, neurogeneticist doing this work from a business perspective, but it takes engineers, it takes biophysicists, biochemists, metrology, material scientists, software developers, web developers, bioinformatics, geneticists. I mean, it takes a huge village of capabilities and expertise to put these systems together. We have complex optics, complex chemistry, complex biochemistry, and lots of engineering and programming. So many roads to Rome. And if it's not building the product itself, we have to figure out how to get that product to market to the customers who need it most, how to run the business, how to keep the books, how to stay out of trouble with legal, how to find the talent and hire in the right people to do all this incredible work. So lots of ways to do that. With that, I will just end and open it up for Q&A. And really, it is my pleasure to be here with you all this evening.
Thank you so much. That was amazing. Okay, I'm going to share my screen again quickly. Oops, I don't know what this is. Okay. So I wanted to just thank both of the speakers again. Hopefully you guys are just seeing my slides now, right? And yeah, okay. And so thank you guys, that was great. I think, you know, I was following along with the chats and it seemed like everyone was really engaged and excited about the talks, and I'm sure they'll have some more questions. I want to thank our sponsors again, both the Southern California sponsors and the national sponsors. And again, if you're going to make some of the DNA, if you finish it at home, we'd love to see a picture of it and try and use these hashtags so we can see it. Okay, and with that, I am going to say goodbye to the people who are watching us later on YouTube. So, but the people who are watching this live can stay on now for a Q&A.